Great. Okay, wonderful. Well, welcome everyone to this very special alumni Zoom. We're so lucky to have Dr. Fernandez-Sesma, who's a graduate of our PhD program and a superstar scientist in our microbiology department. I thought we could go around the room and introduce ourselves, and then you can tell us about your path. How's that?
Sure.
Okay, shall we do this popcorn thing, or yeah, let's do that. Or how I will call on people because sometimes we forget and then there's a lull. So I'm Talia Swartz. I'm a assistant professor of infectious diseases and physician scientist. I study HIV and COVID in the lab, and I'm the co-director of the MD PhD program. And I am a graduate, so a Mount Sinai alumni, of the MD PhD program at Sinai. And in fact, all of the programs at Mount Sinai, the residency and the fellowship and everything too. And then the vice president of the Alumni Association and helping out with some of these alumni Zooms, which is like a huge fun to be able to connect with our alumni. So I'm very happy to be here. And I see Eva is next on my screen.
Hi, everyone. My name is Eva Chebyshev. I am a first-year PhD student in the microbiology department, and I am currently rotating in Anna's lab.
Great. Jason.
Hi, good afternoon, everybody. My name is Jason Soto. I am the Director of Alumni Relations and work very closely with the Alumni Association, with Dr. Colvin, who is our Dean, Associate Dean of Alumni Relations. And so if any of you have been on any of these in the past, you've seen my face. So I welcome all of you and look forward to hearing from all of you. Thanks.
Excellent. Arturo.
Hello, everyone. I am Arturo from Spain. Now I'm working in the lab of Adolfo Garcia Sastre as bioinformatician.
Wonderful. Thank you. Claire.
Hello. I am a postdoc in Stuart Siofon's lab in the Department of Neurology. I'm working with Irene Ramos-Lopez, which was who worked with Anna. And currently, I started a project looking at different COVID strains and studying how they behave in cell culture. And yeah.
Wonderful. Excellent. And Romel, is that how you pronounce it?
Yes, of course. My name is Rommel. I am from Venezuela. I am a postdoc fellow here in the Department of Microbiology, also in the laboratory of Adolfo Garcia. Very pleased to be here and listen Anna to talk about.
Wonderful. Thank you, everybody. Okay, Anna, please tell us your story.
Okay, so my name is Ana Fernandez-Sesma, as you probably all know because you're joining this call. And I am now a professor here in the Department of Microbiology, and I'm also a professor of medicine and infectious diseases because I have a secondary appointment. And I've been in Mount Sinai my whole adult life. So I initially joined, first my lab works on viruses, and I have trained in influenza virus, dengue virus. I work with Zika virus, HIV, and a little bit SARS coronavirus, but most of my lab is really working on dengue and a little bit of flu. So yes, I am very interested in looking at how viruses manipulate the immune system and are able to establish infection in the host. And I really like to understand how the different viruses do it with different strategies. So I started my career in Mount Sinai because I came with my husband, Adolfo Garcia Sastre. We came right after we got married in Spain, and he got a position here as a postdoc. And I just finished my bachelor's degree, my master's degree in biological sciences. So I was really not sure what I wanted to do. I was thinking maybe I can do a master's degree while he's here as a postdoc. Maybe I can get something done, and we'll go back to Spain in 2-3 years. We'll be fine. We'll be back with our family. So it was interesting because it's a funny story that Adolfo didn't really speak English very well. So his English was terrible. So I could speak English. So at that time, believe it or not, there was no internet. So we had a letter from Peter Palese that had invited Adolfo to join his lab as a postdoc. So we arrived and we came to say hello to Dr. Parese that we didn't know. And we came with the letter of invitation. So I came with Adolfo to say hi also, but also to help him with the translation because I thought he's going to be not able to carry a conversation. He could write it, but not speak very fluently. So we go there and they start talking about everything with difficulties. But then he turned to me and said, "So what do you do, young lady?" He still, to this day, he still calls me young lady. But anyway, he asked me and I said, "Oh, I'm a biologist. Can I work in your lab?" So could I work as a volunteer? So he was really good timing because he told me that, yeah, if I was interested, there was a postdoc that actually was going on maternity leave and maybe I could work with her and then kind of continue some of the project when she was out. So it kind of was a little bit like serendipitous. And then I joined the lab as a volunteer. But then after a few months, actually that postdoc finished and went back to her country, and I was able to continue with the publication that she had put together, and I finished all the experiments. And then Dr. Palese asked me, why don't you apply to the grad school, instead of just going to stay here or go to a master's program. So I did apply to the grad school and I got into the program. So but then I just did not want to stay in the same lab where my husband was working. It was a little bit awkward. And I actually realized that, yeah, the molecular aspect was interesting, but I always wanted to work with the immunology component. I wanted to know also, like, if you're working with an animal system, is this actually killing the animal? Is it causing disease? I just wanted to go beyond the viral proteins or understanding some of the more molecular aspects. So at that time, there was still, I mean, he's still here actually. Tom Moran was working, was the only one in the whole department doing immunology. So I asked him if I could join his lab and then really do kind of in between experiments. So it was like viruses that were generated in the Palese lab, but then working with animal models that Tom Moran was developing. And so anyway, I just started to work in this thesis project, and I really did a lot of animal work. But then I realized I didn't really want to work with animals, and I really didn't want to, to do my whole career working with animal models. But during my postdoc, I decided to really test vaccines. Oh, hello. So I decided I really wanted to understand, like, how can you really do this, what is really the concept of vaccination. So we, using different animal models, and different viral vectors, we wanted to understand how can we mimic an infection without really causing the disease. So I really started being very interested in viral vectors and in looking in the animal models initially, but then I wanted to know what are the cells that really actually are important for vaccination. And I wanted to work with dendritic cells, which are cells of the immune system that really are involved in antigen presentation, but also they get activated by viruses. And so that kind of moved my interest into these dendritic cells in the context of influenza, but they are not really the main target for influenza virus infection. So then when I finally decided to really start my own little project, I mean, there was a lot of influenza research in the micro department. So it was really, for me, a little bit difficult to decide what to do with the rest of my career, because I say I'm competing with these big people that are really successful in influenza. So I have to hide my own story if I want to really make my name. So because I was interested in immune cells, I knew about dengue virus, which is very neglected tropical disease, but is affecting many areas of the world. And it happens to infect dendritic cells, which are these immune cells. So I thought, this would be actually a good thing to do, because if I have blood that is from the blood bank, I could actually isolate these cells, and then I could look at how the virus interacts with them in their primary cells, and I can look at the virology and the immunology. So I actually did a little pilot project, and I got very interesting data, and I consulted with my mentors, and I said, should I send an R21? Should I send an R01? And Dr. Palese said, no, send the R01. You have data, send the R01. And I sent my R01 to NIH, and I got funded. So I was at that time like a senior postdoc. So I was able to then go out and interview and for a faculty position, because I thought maybe there is no room for me in Mount Sinai. So I got two different offers outside of New York, which I would have taken, but I love New York. I really didn't want to leave New York, but I thought, this is a career move. I might have to do this. And I told Adolfo, I'm going to interview outside. And he said, of course, do it. But the only requirement for me is that the place has to have BSL-3, because I need to be able to work with viruses. But so I only interview in places that had BSL-3 or BSL-4 containment. And I got two offers for faculty, but then Mount Sinai realized that they probably wanted me to stay. And they asked me if I wanted to leave. I said, no, I actually did not think I could have a position here. So I was able to really get a position here in Mount Sinai. I was really not familiar with this negotiating system that occurs here. So it was all very new to me. And I was very, very kind of surprised by how it works, but it worked out for me. So although many people have tell you, you are not supposed to stay, you're not supposed to stay in the same place, it worked for me. I had two children. My two children were born when I was still a postdoc. Um, I had to make it work with my life. So I thought, okay, this is what I need. I need to be close to my children. I need to be able to go home every day and cook and still have my career. So this is what made my career possible. And this is what made my life as a mother, as a wife, as a friend possible. And also I loved living in New York. So I thought, look, I'm going to give it a try and see what happens. So I was able to maintain my funding. And dengue, it was really kind of being in the right place at the right time, because the dengue field was very kind of young and new. And there were a lot of epidemiologists and people that work with mosquito vectors, but nobody was really looking at these more specific interactions with the immune system or trying to work with primary systems. So I was really not stepping on anybody's toes. I was not a threat to any of the big guys or big people in the field. But it was really, I really made very good connections in the dengue field. And there were a lot of women for some reason, which was fantastic, because women tend to really be friends with women. And so we are a really wonderful network of women virologists in the dengue field, that we really kind of push the field together because we complement each other. And it was funny because at some point someone told me that the dengue is a mafia of women. I'm like, we have one field and it's a mafia. It's like all the other fields are a mafia of men. We just happen to have a field in which women feel welcomed, so it's not really a mafia. It's really a comfortable place for women. So, it's been a great opportunity. And for example, one of these women that is super successful in dengue, that did a lot of epidemiology and had a cohort in Nicaragua, I was able to put together with her a big consortium that it was funded 6 years ago, that we are working on the progress report today. She just sent me an email. So Eva Harris, that she's really a big name in the dengue field. So we were able to put this grant together, work together with her cohort and my approach. So it's one of these examples in which just working together and not competing with each other, you actually make the field better. And it has worked very well for us. And I'm super happy to still be able to work at the level I'm working now, because I started doing things in a much more like smaller scale in the lab, but now being able to be part of this big consortia, you really now have access to human samples, or you have access to technology that you could not do by yourself in your lab with an R01. So you really have to be, so I really think that has helped me grow as a scientist and also as a person, to be able to interact with different people that are from completely different fields than mine. And with also being in this department, I would be an idiot if I don't do influenza. I trained my whole career in influenza. So I was also very fortunate to really have the opportunity to use influenza virus, because it doesn't even target these immune cells for infection, but these cells are they sense this infection with influenza and they shape the immune response in the lung. So I really decided to focus on that. How do these cells react to different viruses, different types of influenza viruses, and can I really understand this response in the immune compartment, not really in the lung? So I really I tried to combine my two loves, which are viral loves, influenza and dengue, but influenza was my first love. I really have very good memories, but you kind of move on and you move to your main love, which is for me, dengue is a fantastic kind of question to a virus in which you can answer many things about the role that immune cells have, but also why there is people that have asymptomatic infection or severe infection, and it's a pediatric disease. So it's, and also in the sense of One Health, what is the importance of this climate change, the mosquito expansion. This is a virus that is transmitted by mosquitoes. So what is the component of the mosquito? So it's really a super interesting virus because it gives you that whole knowledge of many different components that, and now with SARS-CoV-2, were very surprised about these very different manifestation of the diseases. There are many viruses that we know that 80% of the infections are asymptomatic. So this is really not new. But like people didn't know because they're more familiar with viruses that cause a lot of disease. So but there are many areas in the world in which these viruses are endemic, or other infections, and it's not killing everybody every year, but it's causing significant morbidity, and it's really a problem for those countries. So, I think I talk too much, but this is basically how my career developed. And then I was also, one important part of my career has been mentoring. So I really did a lot of teaching. I started teaching when I was in grad school. I really was TAing, and I love teaching. And I was asked to be the co-director of the microbiology training area a few years ago. So I ran that for 10 years. I just stepped aside. I don't like to call it step down. I stepped aside 2 years ago because I was too busy with other things, and I thought they needed also fresh blood. But I think it's something that for me is very important, like mentoring, training, and not only in your lab, but being able to really help other students, postdocs. Now, actually, of course, I stepped aside on the training area, but now I'm the mentor of the junior faculty in the department. So, like you will also need to help them to kind of decide what to do next. So this is really an important part, and I have been also involved in revision of NIH grants. So I've participated in study sections, and for the last 3 years, I've been part of the NIH Council. So I really like to be very active in terms of not only my research, but really other things that are needed in the community, because it's not just being a research scientist, you're also an educator, and you're also, if you're funded by NIH, you have to give service back to NIH and to the investigators. So that's how I like to spend my life. And then I have two beautiful and wonderful children that are now in college. My daughter is graduated. So I did also, I was able to be there for them and at the same time develop a career. So that's, for me, is the most rewarding part, that my daughter is proud of me, my son is proud of me. I was present, but I also had my own career, and I had to travel, and they understood. So I think it's important that you tell your children that this is also part of life. Your mom is not going to be there every day, but it's there somehow. So yep. So that's basically what I would like to tell you about myself.
That is beautiful. Thank you for sharing. And we can open it up for questions if the group has. Otherwise, I can start us off, but I'll give a moment.
I mean, this one is pretty general / basic, and probably gets asked at every Women in Science centered event, but really, you are an incredible balancer, right? So how did you still raise your children so successfully while also being a powerhouse in the microbiology field? I know you said Eva Harris is really well known in the dengue field, and she is, but even before now, when I thought dengue, like I thought you were like the dengue woman in my mind. So
Thank you.
like,
Thank you. No, I mean, I think it's just, it depends on what are your expectations and what are your goals, you know? And for me, I surrounded myself with wonderful people that helped me, you know, and I had a lot of friends, most of them women, that they actually, a lot of them didn't have children. So I could travel. And if my husband was not here, which was 50% of the time, I had my network of women taking care of my kids or helping me. And I had a wonderful nanny that she will stay at home if she had to. But I really built a network of people in which I could rely on because my family wasn't here. But at the same time, I had to, you know, I tried to take turns with my husband to travel, but you take time from other things that other people might do in their free time, you know, so you really try to balance. But at the same time, for me, it was very important. So my career was not about how many papers I could publish every year or how many things. I tried to do my best, and if I was successful, great. But I did it at a pace that was compatible with my life. And then I was extremely fortunate to be surrounded by good mentors, you know, and I have to say I had wonderful mentors too, including Adolfo, that is my husband and my mentor in a way that I can't ask him anything. But Dr. Palese has been fantastic because he's very tough. And I always joke with him because he says, you know, I say, you're pushing me, because he said, no, you can do this. You can do this. Why don't you apply to this? And then I said, you're always pushing me to do things. And he's like, no, I'm not pushing you. I just put you at the edge of the cliff. And you choose if you want to jump or you don't want to jump. So I think those are the type of people you need to surround yourself with. You know, you need people that believe in you and tell you. And I think for women, it's normally hard to think, you know, we have this imposter syndrome for everything. When people tell me, oh, I read all your papers, and I'm like, my papers? You read my papers? So you always think that how come people know me or whatever. And I think that's kind of something that women do. But I think you need people to keep telling you, no, you know, people know you or whatever, you know. And I think it's great that you have to accept the challenges. And if people tell you, why don't you apply to this, instead of saying, no, maybe I don't have time, it's like, okay, let's apply for this. So it's really this kind of mentality, but identifying people that will put out a word for you and also will kind of push you a little, you know, a little bit out of your comfort zone and believe in you, you know. So I think I was extremely fortunate. But I also think I have this kind of way of identifying the opportunities that maybe some people don't pay attention. And I don't know, it's always been like that for me. And I don't know why, if I see the opportunity and then it works out. But I think I have had many failures and I have had many things. So one of the main things in science is that you need to accept failure, like it's not something that is personal. This happens to everybody. So you have to live with constant rejection and constant failure. And if you learn that, then it doesn't get to you. I mean, of course, when they reject your grant, you feel like a failure, but then you're like, oh, you know what, I'm going to apply again. And that's something that is a constant in science. So I think you have many more failures than successes, and you just have to really understand that this is the way it is, and it's not something that you're doing wrong or that you're not worth it or that you're like less smart. So you have to kind of like yourself in a way that is like, this is what I am capable of doing, and that's it. You know, I am a marathon runner instead of a sprinter, but I'm happy with that. Yeah.
Thank you for sharing. One of the things that you talked about was how your career has taken you with different options, but that you've stayed at the same institution, and being in this environment that's heavily saturated with influenza people and being able to carve out your own niche. What kind of advice would you offer in terms of whether to stay at the same institution and how to carve out your own niche, given all of these different competing factors?
Yeah, I mean, it's very difficult to predict because each person is a universe, each department is a universe. But I think my advice would be choose something that you really want to do, and then see if you could do it there, if you will have sufficient support, in your department that even though if it's a completely new idea or new approach, you will feel that you can have people around you that you could go for advice. So it was a different virus. It was not really, I wasn't working in fungus or something that was not, people were not familiar, but at least, but I was the first one working with flaviviruses, which were not in our department. But at least I knew that I could talk virology to people and I could really ask for advice and things like that. So but the main thing is choose something that you really want to do and that you really want to learn about. And then if there is nobody in your department that does that, you can still reach out to people somewhere else, but you might feel that your department is supporting you in different ways. But if you really feel that you need to be surrounded by people that do similar things that what you do, then just move and go there. So it's just wherever you can find the support that is either emotional or like intellectual, or maybe to have sufficient like starting up funds, because you still need time to put together your grants and you really need to move to a place in which maybe you have a better offer. Normally they offer better things to people outside. So like recruitment is actually always better than retainment, so it's not that they're gonna really constantly telling you we want you to stay here. So I think sometimes is a good career move. You might be in a point of your career that you think you need some other collaborator. You really don't feel that maybe your department is giving you, maybe you need to take another direction, and another place in which there is some other person that works on that, and you need that resource of that technology. So maybe it's a good move for you because that will help your projects. And I think it's more make it fit for your career, but also you have to be happy with the people that are in the place that you work. So if you feel that you can go to any office in your department and talk to anybody, I think that's a good place for you. But I think there are other people that don't really need networks as much. I think I am a very people's person and I need to be happy. And I tell people in my lab, even when they come, I say, I like people that like Mondays. I like people that like to come on Monday because they're excited that we're going to do something that week. So I like to come I like Mondays, and maybe I'm weird, but that's what I like. Being surrounded, not just in my lab, but in my department. So I think and you have to be the one that knows if you are happy to be in a place on a Monday or not. So it's very personal. Yeah.
I have a quick question. If you had to do it over again, is there anything that you would do differently? Or would you still stay in academia? Would you consider a different route?
I did consider a different route. Actually, at the end of my postdoc, there was a position in a company here in New York, and I thought, oh, okay, I'll give it a try. Maybe one of us has to be in industry. And I interviewed, and I got an offer. But then the same week that I had to tell them yes, I was told by NIH that I got my postdoctoral fellowship. So I said, oh, I'm staying. So I did stay. But I think I could have done something else. I think I made this work for me, but I could have been happy somewhere else as well. I just think that you have to be consequent with your decision. And when you make decisions, it's because at that moment, it was what you had to do. You can't really say, oh, I should have taken this, or I should have done that. At that moment, it was not the right choice. So there is always time to move again, but I think I don't know, because I am happy where I am, and I don't know if I really would have been happier or less busy, but I'm okay. I think I'm okay. But I could have potentially done something else and not being totally frustrated. There are many things that you can do with a biology degree that you could have been okay. Yeah. I could have been teaching only, for example, or I might end up being doing teaching if I lose my funding. So I think there are many things that I like to do that if I have to change a little bit the approach, I will still be happy. So I don't want to be unhappy. So if it becomes too hard, I'm like, you know what? I don't care about grants anymore. I'll just do teaching or I'll do something else, you know. So
Yeah, then we need more puppies, I guess.
yeah.
Yeah.
And it's very, random, like NIH funding, you could be lucky, but my last grant, I had it for 10 years and then I submitted it and it was not even discussed after 10 years of research and papers and stuff. So you're like, okay. So then you really need to reinvent yourself and say, maybe I don't know, I'm like not giving it the. So I think sometimes you really need to sit back and think maybe I should think it over, but I don't know. It's not always peachy and and great. Yeah.
Right now, I'm thinking so much about you said about the niche, because in the past, I'm related with that because in the past I was working with dengue, Zika, in Mexico, and then here working with SARS-CoV-2. And then I keep thinking, should I try also influenza? But what part of virology? I love virology, I think, but what part of what I'm going to do in the future, I don't know. I keep thinking, I think uh, that part takes time. Right now, we are very busy doing a lot of things in the lab, and sometimes you can't find the time to think what you are going to do in the future. But I don't know, I kind of related what the all the things you are saying about that, and I keep thinking, I'm
I mean,
thinking.
I think things kind of fall into place for you
Yeah.
normally. An optimistic by
Yes.
nature. I'm super optimistic.
Yeah.
Yeah, like dengue is a positive strand RNA virus, as you know, and flu is a negative strand RNA virus. So I always say that I have to work on dengue because I'm too positive to work on a negative strand RNA virus only. So I mean, but I think if you have the experience with virology, you really will. When I wrote my grant on dengue, all my training was in flu. And that's why when I went to talk to Peter and I said, Peter, I don't know if I can send this an R1 because I've never worked in dengue. But I think I can do this because I have data that what I did in flu and I'm proposing things. And so then he said, you know what, you need to try, and then if you can convince them, yeah, that'll be fine. But if you have a good attitude and you sound assertive that you can do it, they might like it. And when the reviews came back and it had a good score, and then he said, bring me the reviews, and say, see, like they never even questioned that you had not done dengue before because you have the virology background. So you really need to spin, you need to spin your own kind of skills or your own research for your own
Okay.
goal, and Tom Moran used to say that I could make, I cannot say it in public, but crap look like gold. But I said, no, I don't have crap, you just don't see my gold. so it's like so it's like having a positive spin on what you do. So if you're trying to cross to another field, just say, yeah, I understand, I'm aware, I have never done it with this virus, but I can, I show you a little bit of preliminary data that I can grow the virus, but I did all these things with flu that I could apply, and maybe this is a better system. So really give it a spin, but not everything you do is actually part of your baggage. So use it because although it might not seem related, it might be related. You need to make the connection and say, this is going to help me now solve this problem because I have experience on this. So you need to convince the reviewers that you're capable of doing it, and use what you have as experience, but it doesn't always work.
Mhm.
You just have to try. And I also have a lot of friends that went back to their countries and they actually look at what is really needed in my country. So if I really want to go back to my country, what is the main problem there? So my friend Andrea Kamarnik that went back to Argentina, and she had worked with polio her whole life here, I mean, her whole career and then she realized that dengue was a problem. So she said, I have to work with dengue. And polio is not from the same family, but it's also a problem. So at the end, you really shape your career based on the needs in your country or the needs in the university that recruits you. And you can really, make it work.
I was wondering, and I mean, you're just always a great example. It's like every time you talk, like obviously you are super compassionate and soft and nurturing. And I was wondering how you hold and optimistic and positive. Like, how do you hold on to that in a world that just constantly tells you that in order to be successful, you need to be tough and cutthroat and competitive and like I just really admire how you're so able to stay true to yourself and be at a position of power. Like I think that is so hard to do. I can't even conceptualize like how to do that.
I don't know if I have a position of power. I think I just did not want to become successful. That was not my goal. My goal was to keep working on what I liked, and it worked out for me. And because I've been fortunate, I am positive because I know it works, and I think for me, it was more just not to have the wrong expectations, and someone told me that I will never be successful in science because I was too nice. And I said, I'm going to prove them wrong. It's like, I didn't think I had to be nasty to be successful. So that's the thing that for me, that was not my goal and my ideal. So I said, maybe I will not be successful in science. I don't care, but I cannot be nasty so I can be successful in science. So I built my career to do something that made me happy. And if it was a success in the sense that I could have people in my lab, train people, and those people had opportunities to build their careers, that was great. Because to me, that I'm very proud of have trained people that now have their own groups or that are working in industry or working or teaching or what. So they, I am a platform for them. I was a platform for them to get to their next step. And that was my goal, to really be a place in which different people could come, be trained, and then be successful in their own fields or in their own places. So if at the same time I was able to maintain my funding and be able to get opportunities to participate in council or doing all these things, that was great. That's icing on the cake. But I also think many people could have had that opportunity, but they were too afraid of taking it. But it's just this fear of failing, I think that but yeah, I don't think you need to be nasty. It's just but I don't know.
I mean, you proved that, right? So yeah.
I don't know. I'm not used to so many compliments.
I'm sorry.
It's like it's a little, yeah. No,
I'm
it's
so
funny.
sorry if that's weird. I just
No.
I
I'm
really
just
admire
kidding.
you.
I'm kidding. Yeah, but thank you. I appreciate it.
Can
No,
you tell us? Oh, sorry.
yeah, no. I was just gonna say that I think we have to stay away from stereotypes and things like that. So I think, I think that's the main message. You have to, you are what you are, and you have to make it work, and then just not have these completely unrealistic goals. And that's because normally is the problem is the expectations are completely wrong. So that's when people are unhappy and they just feel like a failure because their expectation was maybe too high or maybe too was not really the what could be obtained by that person because of other things going on in their lives. So
Can you talk a little bit about how COVID has impacted your life, career as a scientist studying viruses and the immune system?
I mean, COVID, when COVID started, we were actually contacted by NIH because I had this consortium for dengue that I could do a lot of different analysis, phenotypes of cohorts. So they told us, can you please use what you have and then now study COVID? So we said yes, absolutely. So that's why we started working. But it turns out that COVID doesn't infect the immune cells. COVID didn't infect all the systems but I tried and we did a little bit of some experiments. I think hopefully we'll have some manuscript. But I realized early on that this was not going to be like, first of all, I didn't want to be in the rat race of like being publishing every 2 weeks and things like that. That doesn't go with me. but I was happy if I could contribute something, of course I would. But I think I did not really jump into the COVID train to really be full force. I have to like live but somehow I found myself very, very involved with COVID because of science communication. It turned out that a lot of people came to me and asked me to explain. And in Spain, I was asked to be in a lot of interviews and uh, things like that. So it was really, so then I felt like I was doing my part. You know, I was helping people with vaccine hesitancy. I was helping people with like things that were circulating that were not true, and trying to explain things a little bit on Twitter or like in interviews or in the TV. So I did do part of that, but my life changed in the sense that there was this kind of pause in our research. Then we were able to get back. So at least the lab was not closed. So I was happy that we were doing something, or we felt that we could do something. But in my life, my kids came back home. They were in college and they came back home. So we became a full nest. But then my daughter graduated during the COVID. So it was totally like anticlimactic. So she closed her computer and said, "I'm done, Mom." So she was like, "Now what?" But she did film and animation. So actually she's been working nonstop because there's a big demand for animation. And she actually did a film and she won a Studio Academy Award. So she was like super, so she's been like it's big actually the year of her life. But she's home and she cannot hang out with her friends. But our son came back. So we had a completely different dynamic. So we had to live in a different house because we had not had our kids at home. But it was great because we came to work, we were working nonstop, but then the evenings was like, now our kids are back and they're adults, and then we can do things together, like watch things, cook things. So it was really actually the best part of the pandemic, to have our children back. And we actually really were happy to be together. But then our son went back to college in the fall, but our daughter stayed. So it's really changed in the sense that we it wasn't as bad as we thought it was going to be. You know, it wasn't as bad for my daughter not to be able to look for jobs because she could work from home, and our son was able to go back to college. So, and we keep working. So I think it was, being impacted in the sense I couldn't go to Spain, I couldn't visit my family, but I think, and I'm happy that the vaccines are here, and then now our horizon looks a little bit better, and then this is gonna end soon. But it did change. It is really like. And sometimes it was overwhelming, because like everybody, and at the beginning with the lab, you don't know how all the people in your lab are doing. I wanted to make sure they all felt connected, so we kept having lab meetings. So people that moved back home, we were meeting every week, and the four that were in the lab that were doing some SARS-CoV-2 work, they were telling them everything they were doing. So we tried to keep it connected, so they felt like, okay, the lab is doing something, So yeah, it was hard to really try to keep everybody engaged and make sure they were doing okay mentally, and and they were still excited to come back. But yeah, it was very hard because you have to make sure people that depend on you are taken care of, and that was, yeah, a little bit hard at the beginning. Did I answer everything? I don't know.
So we have about 10 minutes left. So are other questions from the group?
I mean, one thing I can tell you, I don't want to work at home. I can't concentrate at home. I find so many other things I have to do. It's just hard for me to really concentrate. So I totally admire people that work at home and can get things done. I just get much less done when I'm at home somehow. But you have to do it, and you do it. We are fortunate that we live 2 blocks from Mount Sinai, so we could come even in the hardest times of the pandemic. So we could come to work, and that also was helpful for us because our kids were back home, they were in class, so we all had our we were separated during the day, but then we kind of met, so we were not on top of each other. Because for them it's hard, you know, they were not living at home anymore. So coming back to your parents' place and being a, you know, like young person is really okay, but sometimes not that much okay, you know. But yeah, so we made it work.
I have one quick question.
Mhm.
So, Anna, you've had a very impressive career.
Thank
And
you.
is there anything on the horizon that you still want to achieve, or is there still keeping in mind the next couple of years?
Hmm, that's interesting. I don't. Let me see. Let me see. I would like to. I think I'm very grateful of where I am right now. I don't think I want to achieve anything in particular, but I wouldn't mind having a position that was more kind of demand in the administrative part, like some that I will be taking care of, I don't know, chair, vice chair, something like that. I don't see that in my near future, but it's something I would not say no if I had the opportunity, or I would consider at least, because that I think I can do things in which I can deal with people and I can make decisions and help, and especially changing environments that are not friendly to different things. So that's why I keep being in many committees, trying to change things from inside. But I think I could still do something important or helpful if I had to run a big group that was not just my lab. But I don't know. I'm not gonna actively search for that, but that's something that maybe by the end of my career I would like to have something like that, a position with more leadership in, but not just your own science, but other things that you could. I mean, I did a little bit like the MTA. I was running the training area. I mean, I've been kind of flirting with that in different places because I really like doing that. I think that's an asset that I have, that I can do it. So I like things that will actually enhance that, and maybe in the future I will do more. I will have more administrative duties, but maybe not. Maybe I will just stay the way I am. Yeah.
And can you speak a little bit about your Mount Sinai experience for our current Mount Sinai trainees and graduates, about what you can point to that Mount Sinai has given you that you think may be unique based on your experiences with other institutions?
I mean, I think for me, Mount Sinai is very different Mount Sinai's. You know, there was a Mount Sinai when I was a student, and the school was kind of like small and cozy. We all knew each other. It was very, I don't know, it was more, I mean, there were not big MTAs at that time, so you were in the departments. So it was a little bit more maybe academic. I think then when in my postdoc, I was a little bit out of it because I had two children. It was a little bit difficult to kind of interact with the community. So I think my postdoc years were the least connected to Mount Sinai. And I think that also shows that postdocs are a little bit, they don't belong to a group. So it's kind of the time in your life in which you can get more disconnected. But then as a faculty, I really liked it because there were many junior faculty that we became friends, and we are still friends. And I kind of, it was exciting building your lab together. And I had to be in a different department because there was no space in micro. I had to move to a different floor. So I was always like opening new territories. And I was very excited to really interact with other people in different departments. But I think Mount Sinai also gave me this opportunity to now look more into the clinical aspect, or like how to connect to the clinic. Unfortunately, there is not that much dengue in New York, or fortunately. That's something I wanted to build a little bit more with like Dan Kaplivsky. I really talked many times, and to see maybe we can do something. But it was really like trying to discover new things. And Mount Sinai has allowed me to go from like very basic science to a little bit more translational. And I think, like now the relationship I might have with people in infectious diseases or in other departments that are more clinical is completely different. So, and also being in Mount Sinai for so long, the grad school, Mount Sinai, everybody knows you. So I've been in so many different committees it has been great in terms of learning, like learning how to do different things that are not necessarily in your area of expertise. So yeah, I recommend it.
And if you could go back and do something different, would you change anything?
Maybe I will change, I think being a little bit more vocal at the beginning about women's needs. That I don't think I was as vocal. I took things because I was told that maybe I shouldn't be looking for a postdoc outside if I was pregnant or things like that. So maybe I would have been a little bit more or less afraid of doing things. So I'm not saying that I stayed, but I kind of stayed because I was afraid of looking for something outside at that point. And maybe, I mean, it was the best thing for me because it was more manageable, but I didn't push myself because I was afraid that I was going to fail. So that's something that maybe I would have tried a little bit harder, but yeah, I tried not to be, because I think you always have this association with like angry women complaining. So I tried to stay away from that. At that time, I thought if I speak up or if I ask too much, they're going to say, oh my God, she's difficult. And I did not do enough, So I think I could have pushed a little bit more the envelope in certain places, but now I would have done it differently, probably. But I'm older and wiser.
I think it's easy for us to beat ourselves up, but this is a learning process, and you've acquired these skills and you recognize your worth and value and all the amazing things that you can achieve, and that has given you leverage to better advocate for yourself. So I think it's hard. We can all push ourselves to be better, but I think your story is so illustrative of how you can build a career within this institution by sort of leveraging resources and mentoring and carve out your own niche in a way that's so beautiful and sort of holistic.
Thank you. I think you have to stand out. And I also have to say, I am married to Adolfo. So you're also considered like someone else's wife, someone else's mentee, someone else's, and you've been here also, you were someone's student, so then we are kind of forced to always have our own identity. And no, I'm not someone's wife. I was told once, and I got very kind of upset because someone told me, oh, behind that great man, there is always a great woman. I'm like, no, in this case, it's next to. So you have to keep saying things like that and saying to people, correcting people, because I say, no, I'm not behind. What do you think I'm behind? No, I'm not behind. But it's just this kind of, you have to push yourself all the time because you have to demonstrate all the time that you're worth it, that you're not here because you're someone else's wife, that they just retain you because because that person they didn't want to lose. So you have to push it, because if not, they I don't know, you just don't want people to perceive you as like you're just a package that came together. So yeah, so I think that's something that in Mount Sinai I had to do, and maybe that's what pushed me to be in the grad school. So I wanted to do things that were different and were my own, and people knew me because of me. So
wonderful. Well, with that, I would like to thank you for all of your insights and sharing your story. And
thank you for inviting me. Yes, and you should have talked more about your career, because you are also a fantastic example of like doing everything and doing it well.
No, I don't know. Well, we have a beautiful network of Mount Sinai alumni, and to all of our current trainees, you will join that as well.
Yes, of course.
Thank you so much, and have a wonderful evening, everyone.
Thank you.
Okay. Thank you.
Thanks, Anna.
Bye. Thank you. Thank you all.